So I'm very, very proud and glad to have Michael Sixt here from the ISTA in Austria. Michael is in the medical faculty, they would call it an Erlanger Gewächs. So he's deeply rooted in Erlangen. He studied medicine here, did his PhD basically here. I think you also worked in dermatology or something for a bit.
Mm-hmm.
Then he went to Lund in Sweden for a postdoc. From there to Martinsried, to the Max Planck Institute as a group leader. And then he was one of the founding members of the IST, right? Like in 2010, where he has been since. And since then, he has been working on immune cell migration in 3D, 2D versus 3D. He has made many, many breakthroughs in the field. Some of his papers I mentioned in most of my lectures. And so I'm very, very glad to have you here, and I very much look forward to listening to what you're doing right now.
Okay, so thank you very much for the invitation. Thanks a lot for the introduction, Christian. So it's fantastic to be here. For me, that's very nostalgic because I did my MD thesis actually right here at this spot. So that was the red brick building. That was a bit longer. So you demolished it for this place here, which is obviously turned out fantastic. So we really already had a very, very nice day with very nice discussions. So I'm going to talk about the main interest, my personal core interest that I had actually since I started here in Erlangen to work on immune cells. So this was always we do a lot of other projects, but one main focus is really how does the single cell move and how does it deal in its complex environments. And I'm going to tell you that the first part is something that has been published this summer. It's basically as an introduction. The second part will be a bit more wild, very unpolished and a lot of preliminary things. So I hop right in with an experiment that Jörg Renkewitz, which I think he was already here visiting a few times. So Jörg did when he was postdoc in my lab. This is an assay where we put a confiner, so two parallel surfaces intersected by pillars, and then we put dendritic cells here at the starting line, and they are attracted by a gradient of the chemokine CCL19. So they're very good in migrating up this gradient. And the point is here that we have a pillar forest that has dense spots and not so dense spots. And what we can see when we look at the trajectories of the cells is actually that they nicely move up the gradient, but they kind of avoid the dense spots. Sometimes they have to pass through, because if they hit them head on, then they have no other choice, then they go through, and then they're going to get a bit slower. And this you can see when I start the movie here. You can see all the cells are moving up. And so they're usually fast when they bypass, but whenever they hit the dense area, they have no other choice, and then they go slower. So they do kind of a trade-off, the directionality of the gradient versus the geometry of the environment, and they always choose the fastest path. So you can also see that in more complex settings. So here we put the cells in a three-dimensional collagen shell. So they are also moving up the gradient. And this is a blob of the shell that has higher density. And what the cells are doing is that when they hit it tangentially, they move around the dense spot. When they hit it head on, they move through. And then you can see it in the color coding. They get slower in their migration. And this is also what they tend to do in vivo. So they always move kind of the path of least resistance. We found out, not in much detail, but we got a feeling that the main decisive factor here that makes the directional choice is actually the nucleus of the cell. So, which is, I think, quite intuitive, because the nucleus seems to be the most bulky part of the cell. So they use it as kind of a ruler to navigate through this environment. And maybe you get an intuition for that in this movie. So here we again have a forest of pillars. And there are two paths now, one with bigger pores in orange, one with smaller pores in green. This is the dendritic cell that is moving up the chemoattractant gradient. And you can see that it's kind of, the nucleus is almost scanning these pores. And then they always slide through the bigger pore. So from this, we thought, okay, and like in other control experiments, of course, like the nucleus seems to be decisive, at least in this situation, to pick the path of least resistance in this complex environment. So it's important that the nucleus is at the front, because if it's at the back, it doesn't make sense, right? Then the whole cell would be through, and then they would have to funnel back. So it makes sense to put the nucleus to the very front, and we know how that actually happens in leukocytes. So this is mainly done by the force of ectomyosin contraction. So this is a wild-type cell. You see the nucleus is very much pushed to the front. So if we block here ectomyosin contraction, we can do that pharmacologically or genetically. Then the back gets stuck and the leading edge nicely moves forward. And you can even cut the cell here, and then the cell moves much better without the nucleus because it doesn't have this load here. And I show you some of these examples where this happens naturally, that the cells rupture. So this is coming later. So, but myosin, it's really this is decisive here. It's this toothpaste tube kind of mechanism that they squish the nucleus forward so that it's always located at the front. And this makes this typical amoeboid configuration of a cell. So a cell between two surfaces here, you have the nucleus. There's ectomyosin contraction behind the nucleus, pushes the nucleus forward. And this makes this configuration that the cell move with the nucleus first, and the microtubule organizing center and the organelles at the back. Because here you have the mTOC at the back with all the minus end motors. The organelles are transported there. So if you look for lysosomes or Golgi or so, that's also at the back of the nucleus. So it's typical for fast leukocyte. So classically in the textbook, it's like this. Amoeboid migration has the microtubule organizing center at the back. Cells move along the path of least resistance. They don't digest their way, whereas a mesenchymal cell has the mTOC at the front. And especially Philippe Chabier in Paris provided very good evidence that this location of the mTOC at the front is necessary, that the microtubules target then to the front, where they deliver then the proteolytic vesicles that then digest the matrix so that the cells drill a path, basically. But this is way, way slower, right? This is up to 100 times slower than a leukocyte that just moves wherever is space. So this is really the slow version. And this also includes very tight adhesions to the substrate, whereas this is not necessary in the amoeboid migration. But I showed you in this experiment before that if the cells hit the dense blob head on, they have to go through the dense part. And you can also enforce it by actually putting something interesting for the cells here. So if you put a very steep chemokine gradient here in the middle of a dense area, then the cells are also making it through the dense part. So we were wondering how, if they move then in this slow version here, if they do it somehow differently. And so we started with a very simple experiment here. So we put the cells in a 3D collagen gel and then measure where the centrosome is and the nucleus is. And so they are directionally guided here so that they're all attracted to the right. And we can see in the not so dense gel, the mTOC is at the back, so this typical amoeboid configuration. But in the high concentration, they actually switch position. So then you get this more mesenchymal configuration with the mTOC at the front. So the cancer people would say, yeah, this is a typical amoeboid to mesenchymal transition. So it's a big term, and you would think, okay, maybe there's a lot of signaling involved and so on. This is what the cancer people say when the cells transform and so on. We think it's actually, or we worked for quite a while on it, and we think it's a highly trivial process that you get this polarity switch, and that this is a purely mechanical consequence of the cell being too much confined by the extracellular environment. So I can illustrate that in a very simple experiment here. We put a cell here into a channel. The channel is very narrow here, and then it's an open path. And there's a chemokine gradient in this direction. So in blue is the nucleus, in green the centrosome. You can see that once they enter the channel, actually the mTOC moves to the front. And then once they're in, then it switches position and the nucleus again catches up and leads the cell. So it means it's a very transient state, and that it's actually not a big switch of polarity. It's more like the mTOC is always in the middle of the cell, but here the nucleus doesn't fit, so it falls behind. And then once the cell is free, then the nucleus is being squished forward by the myosin. And this we can also kind of see when the cell enters the channel. So if we look at the myosin light chain here, it's primarily at the back of the nucleus. And so when it's fighting through, and if we block the myosin while it's moving through the open part, then also the nucleus doesn't catch up. They keep moving with the mTOC at the front. So it's like pretty trivial, I would say, the switch of polarity. But it's not only the mTOC that is at the front when the cells move, but there's also an interesting actin reorganization happening. So this is now in a more physiological environment. That's we put a dendritic cell in a collagen gel. You can see the collagen fibers here. This is the nucleus. This is actin. And here the nucleus is obviously stuck in a pore here, and it's trying to fight through. And if you look what's happening during the time where the pore is opening up, there would be the mTOC localized here. It's not visualized here, but it's always there at the front when they move through this constriction. But what you can see at the same time, there's a burst of actin in front of the nucleus. So it's like very tempting then the idea that, yeah, maybe this is, you get this explosion of actin, and this is how the cell opens the pore to make space for the nucleus to follow. So we can see the same thing again in this microfluidic assay. Cell moves through a small pore here, and then you get the burst of actin when it moves in, and this burst is gone here once it's inside. So there's really actin reacts to maybe the compression here, and we can also do that passively. We can push onto a cell with a little micropipette, and it basically pushes back with an explosion of actin. So we got very interested in this structure here. What are they doing here? Are they trying to open the pore, which is, of course, not possible here because it's a PDMS channel, which is indefinitely stiff for the cell. So we were interested in this actin burst here. So we took a configuration where we can look at it more quantitatively. So we put the cell in a simple confiner here. So we squish it down to 4 microns. And here it's not squished, or that's 8 microns, so that the cell is hardly confined. And then if you look from top, you can see that in the 4 micron confiner, you see this blob of actin in the middle. So in the center of the cell that you don't see here when it's not confined. And when we quantify this actin, this central actin structure here, then it's really, it increases with confinement. So the more we squish, the more you have this burst of actin in the middle. You look at it at higher magnification, we can see it here. So it's really the most prominent actin structure in a confined cell. So you have lamellipodial actin here, in white is the nucleus, but the most prominent thing is this central actin structure. So it's not directly on top of the nucleus, but it's next to the nucleus, and it actually associates with most of the organelles. So it's we see lysosomes there. The Golgi is kind of scattered around this actin structure, and in the center we actually have the centrosome, so the microtubule organizing center. So I have to say, in these cells, in immune cells, the centrosome is really the only microtubule organizing center. So if we look at a movie now of microtubules in green and the plus end microtubules here in red, this is the nucleus, then you can see all the microtubules come from the mTOC here, and they radiate outwards. And this is where this actin structure would look like. It's not on the centrosome, but it's in the let's say the centrosome is the center of this big cloud of actin here. So in a nutshell, it looks a bit like this. So you have the cell here that has a lamellipodium at the front. When it's confined, then you also get this actin here. And in the center, you have the microtubule organizing center with the microtubules. And I'm going to come to that later. So very tempting, the idea that maybe it pushes up and down to make way for the nucleus. And in order to measure that, we established pushing force microscopy. So we incorporated beads in agarose and let the cells move under agarose, so that the cell is basically like moving under a carpet. And then we can measure how they lift up the beads. and at the same time visualize the actin. So in red are the beads, in black is the actin, in blue the nucleus. And this is when you look from the side. So we can see that they're passing below the beads, and then we can see when do they lift up the bead. And if we measure that in chymographs here, then we can see the structure that lifts up the bead is actually this actin structure here, and not the nucleus. So when the nucleus passes below the bead is already on its way down. So it really seems to be the central actin seems to be the main pushing structure. That's also quantified here. Again, this actin is not on top of the nucleus, it's next to the nucleus. And if we enucleate cells and let them migrate, that works well. Actually, we can get rid of the nucleus and they still migrate quite well. Then we don't lose this actin structure, and it's still there to push upwards. So we don't need the nucleus for this activity here. So central actin really seems to push up and down. Yes? In this case, it's mainly at the back of the cell. Yeah. So this is when they're like highly confined. If you put them actually under stiff agarose, they have the tendency to move with the centrosome on the front, like in the dense gel. If you put them in a soft environment, then it's rather located at the back. And then you don't have much of a central actin structure here. So yeah, it really seems to push up and down and maybe make way for the nucleus, which would make sense in this setting that I showed you before, that whenever the cell goes through a pore, it opens it up. But now the cell has a kind of a dilemma because it has to push in two directions. On the one hand, the actin has to push forward. On the other hand, it has to make space in the center. And this is something that is, in general, I would say it's not very well understood. How do the cells coordinate on a global level what they're doing? We know a lot how one protrusion is pushing. We know a lot how one focal adhesion is working, but not how it's coordinated on the cellular scale. Yeah.
Any idea how the actin is pushing at this place? I mean, at the front, it's immobilized, but you come.
Yeah, I come to that, but I won't have a really satisfying answer to you. but that's what we are really working on a lot. How this pushing works. Yeah, I try to give a few answers later. Yeah,
but that's not what you are alluding to here.
No, I will.
Okay.
So that's in the end. But as I don't have a final definite proof of that, but we think really it's the microtubules that are involved in the pushing here. Yeah. So that's how I draw it also here. So I first wanted to talk a bit about the coordination. So how does this actin and this coordinate? Because if it's uncoordinated, the cell would have a dilemma. So we first just did cross correlations of the cell. So how does the central actin behave when the cell is moving forward and makes protrusive forces? So what you can see from this movie is that whenever the cell has a run, like now, when things are going well and when it's moving fast forward, and then there's a lot of actin at the front, so they spread a lot at the front. But then the central actin, which is binarized here, is disappearing. So it fluctuates. And whenever the cell has a nice protrusion forward, it has a little central actin structure. So it's really in the sense that if it gets stuck, it has to push a lot up. And if things are going well, then it just disassembles it. So this is by cross correlation, where we can always see, there's a anti-correlation between protrusive speed and lamellipodial spreading and the central actin structure. But yeah, that's all correlative, and we were wondering how can we look at this functionally, and for that we had to find out how this central actin structure is regulated, and I just skip over it. So in the end, we found that CDC42 is the central row GTPase that drives this central actin structure, but CDC42 is doing a lot of other things. So we looked at upstream regulators of CDC42, and we were lucky that we found one GEF of CDC42. It's DOC8 that specifically locates, when we couple it to GFP here, that specifically locates to this central actin structure. And it doesn't locate to the protrusions here. So it's really specifically there. So yeah, red is the DOC8, and white is the nucleus. So you see that it's really mainly here in the center, and you don't see it at the lamellipodial protrusions. So if you look at CDC42, there are also dots in the lamellipodium. So this GEF, it looked like it's really the CDC42 GEF that activates it in this structure here. And when we knock it out, and we can also look at it in these channels here. So when we look at the DOC8, it also accumulates here in the narrow part of the channel. And when we, so it's basically almost looks like the actin in the narrow part of the channel. So when we knock out now DOC8, that was a lucky thing. We had 100% phenotype of a cell that is completely normal, but it's just lacking this central actin structure. So DOC8 knockout doesn't have it. And basically, if you count, it's 100% gone. And if you put it back in with a GFP fusion, we can rescue the cells. So the DOC8 is going again to this actin structure, and it rescues the actin there. So now we had a tool basically to measure the function of this central actin. And when we do this pushing force microscopy here, we can also see, okay, actin is not pushing up anymore because it's not there. But the little rest of the pushing forces, upwards pushing forces of the cell in these agarose assays, they are actually associated with the nucleus. So the rest of the load is held up by the nucleus, but if this central actin is not there. I told you before that this central actin it's the most prominent actin structure in these cells when we confine them. But when we look overall F-actin levels in the DOC8 knockouts that don't have it, this central structure, they're almost the same. So it's not that they have less actin. So that means that somehow this actin structure, the actin has to be redistributed somewhere else. And where it goes is actually, it's the lamellipodium. So it's more at the front. So basically you lose it at the center, and instead they put it to the leading edge. So very simply speaking here, we would say that the central actin structure, it also acts not only like a pushing device, but also as a capacitor for actin. So it communicates with the actin at the front, and if you completely depolymerize it here, if you take these DOC8 knockouts that don't have it, then the actin goes instead to the front of the cell. So the question is, of course, now, what is the effect on the motility of the cells? So we put them in 3D collagen shells where they have to kind of navigate their way through. And then we see that the DOC8 cells, if we quantify here, are way, way slower. So this is here the chemotactic speed towards the gradient. So the cells get really, really slow. The interesting thing is how they look like. So what they're doing is that they really massively get entangled. So the leading edges kind of run away from the cell body. The cell body cannot follow because we think it cannot squish forward. And what happens then regularly is that they rupture. So basically here you have a situation where the cells get longer and longer. So leading edge runs away, and then here, it ruptures. And then you have, at the end of the assay, you have a lot of little fragments in the assay that are moving without the cell body. And if you run this assay for many hours, then the cells are killing themselves by tearing themselves into pieces. So it's really kind of suicide by chemotactic migration. If you don't have a gradient, they also don't rupture. So you kind of have to direct them. They're kind of, the front of the cell is hyperchemotactic. So we can, so this is just the quantification of the fragmentation here, and this is just an overview where you can see in the end all these little fragments here moving around. So they're getting more and more over time. And here you can see a nicely entangled cells where you have all this fighting leading edges. So yes, we think if you take away the central actin, then more actin is put to the front. And this is a problem if the cell body is stuck because the fronts move away. But we wanted to give it a test now. So what happens if the cells don't have a problem with the cell body anymore if we let basically the leading edges guide the cells. So you can do that by putting them in a maximal simple environment. So we just put them in a one-dimensional channel where there's no choice, there's no way they can entangle, and there's no need to squish the cell body forward. And then actually the DOCK8 knockouts are getting faster than the wild types. So if you quantify here, they're considerably faster. So we think this is because they put more actin into the leading edge, and the leading edge can now drive them forward with more faster than in the wild type situation. How this exactly looks like with the actin flow, and so we can discuss. We are not 100% sure yet, but. I think this is something that must be like a six micron channel or something. So at least that it's not a problem for the cell body to follow. Yeah. Mm-hmm. Sorry? Yeah, no, this is actually, yeah, that's a good point. These channels are not passivated. We would really like to do this experiment with completely passivated channels, because maybe they have to push a bit outward to kind of chimney climb in the channel. But yeah, it's not so easy to passivate channels, actually. So we would have to rather get rid of the adhesion receptors here, which we didn't do yet. But that's a very good question. Yeah.
Did you ever try it with like undulating channels where they have more constrictions to push through?
Yes, they have no problem passing constrictions. So if there's one constriction, they can pass through. So the myosin contractility and so on seems to be okay. But the interesting part would be actually, because these constrictions, they're actually so stiff that there's no point for the cell. It cannot dilate them because it's PDMS. So I think it would be different if it's soft channels. That's
what I was thinking. So that there's a trade-off between those two modes, and where you have a flipping point where the DOCK8 actually gets slower because it cannot push through anymore, right?
Absolutely. I think this is just I got very frustrated with stiff channels. So I come to that in the end, okay. Because it's so if the cell moves through a constriction and it doesn't even have the option to widen it, then yeah, what does it mean? Then it's actually just the question how much can it compress its internal compartments, and whenever I think for a cell that has a problem pushing outwards, if it goes through a constriction that is stiff, the pushing outwards is rather a waste of energy and time. So this is why if you get rid of the mechanism, it's getting through quicker. We showed that in wasp deficient cells, but yeah, we need soft channels, so I come to that. Yeah. So yeah, basically with this, we think now we can show something like a coordination of pushing forces in the cell between the leading edge and the cell body, so outward pushing and forward pushing. And then we have the myosin component that is always trying to squish the nucleus forward. So whenever there's enough space, because either it's pushed open or the environment is wide enough, then the nucleus is moved to the front, and they move via this front steering mechanism. But yeah, so I'm not saying that we completely proved that, but maybe we are getting there. So now one important question is actually what happens here. So you already asked about the microtubules here. So what is actually, how can actin push against something if there's not the nucleus as a counterforce here? And in general, I would think the nucleus is not a good intracellular counterforce, because maybe it's actually not so stiff, and you don't want to use it as a wedge, right? Because it's very sensitive. So, but yeah, in this case, it's not even there, and we can enucleate the cells, and the mechanisms that is there anyways. So one idea would be that it's actually the microtubules that push outward here. So we want to know what's happening here, and so how the microtubules are talking now to this central actin structure. Actually, if we depolymerize microtubules with nocodazole here, then indeed we lose this structure, it's much reduced, the central actin structure. I think it's not well visible here, but that's the red spot is the actin. And this is with nocodazole, we lose the microtubules, and we get rid of the central actin. But the problem is also you mess up the whole internal anatomy of the cell. So Golgi is scattered around here. So not a very good experiment, but an indication that there's some type of crosstalk here. So what we are currently trying to do is we try to get better tools on microtubules to more subtly manipulate the microtubules and to look at this crosstalk. And I show you one attempt here. So we started a few years ago to work on a new centrosomal protein called ACNA that has been described by Magdalena Götz. So ACNA is a protein that used to be a transcription factor until Magdalena Götz found out that it's mainly located on the centrosome in this paper here. And so we stained for ACNA in the dendritic cells, and you can see it's really only one dot. There's absolutely no indication of a nuclear localization in no state whatsoever in the leukocytes. So we only see it at the centrosome in red here. And then we knocked out ACNA, and it's nicely gone. So we can delete it. It doesn't affect the differentiation of the cell. So cell division is completely okay. We have ACNA knock out dendritic cells that are also migratory and responsive to the chemokine. But their microtubules are pretty distorted, I would say. So these are wild-type cells, nicely aligned microtubules. These are the ACNA knockouts, and they're kind of all over the place. And if we look at super resolution, you can see here in the wild-type cell, it's all kind of originating from a microtubule organizing center. And in the ACNA knockouts, they're kind of all over the place. We don't even know where they originate. So this is very preliminary, very early. So we have to find out where they nucleate. But at least we cannot see an obvious microtubule organizing center here. Also, if we stain for gamma tubulin, which should be at the centrosome here, so we can see it in the wild-type cells. So you can always see the dots here for the gamma tubulin, and in the ACNA knockouts, it's pretty much gone. So you don't see gamma tubulin at the centrosome anymore. It's quantified here. And now, again, when we look at the actin, then in the ACNA knockouts, it's very much, this central actin structure is very much distorted. So you don't see it in a one nice spot anymore, but it's either not there or completely scattered around. And when we look at the migration phenotype of the cells, then, so these are in a 3D collagen shell here. So these are the wild-type cells, these are the ACNA knockouts. They almost look like the DOC8 knockouts. They get very slow, especially over time here, and then you get lots of fragments in the culture. So basically, you see here in the right-hand movie, you have very often that the cells split in two parts. So it's not like with the DOC8 knockouts are a bit more extreme, that they make multiple protrusions. So for them, it's really the decision point that they rupture very often. So it's kind of an indication for us that now we have basically that we have a functional crosstalk here between the actin and the microtubules. So, but yeah, as I said, we have to collect way more tools to understand the microtubules, and we need very better ways to measure actually these mechanical forces here at the center. And I showed you this pushing force microscopy, which is kind of very unsatisfying because the cells are completely pushed flat. They're under a carpet, they're pushing upwards. So we invested a lot of time, or not we, but my postdoc, Mario, actually, he invested a lot of time because we wanted to make soft channels. So this I took from his lab meeting. This is all the failed attempts. So it was very frustrating. We tried many things like soft PDMS. And so if you make it soft enough, it's always collapsing. So you cannot maintain a soft channel easily with all these approaches. So in the end, he found something that works. And that's based on the good old polyacrylamide. And so with that, we can now make feature casting of channels. So basically, we have a wafer. We have different designs of the wafer, so we can make whatever splitting channels, wide channels, or whatever topology we want. And then we have to put little pads as spacers. We put the polyacrylamide on. You need a cover slip on. Then you take the cover slips, you mount it on a cover slip and put it on the bottom of a migration chamber. And this sounds now super easy, but it was a long way to find out how it works. So sorry to my postdoc. And then you basically can you need the spacers at the beginning here, and then a second type of load-bearing spacers. I don't go into details about this works, but in the end we can get channels that are deformable. And we did a bit of preliminary characterization of these channels with optical tweezers about the stiffness, and we can get around 1 kilopascal. It's pretty soft. And we used optical tweezers to measure that. And this is a movie of, in this case, T cells that are going through the channel here. And you can see, it works. So we have features in the channels like ratchets here, and they move through, and then they get collected in these holes here. We can also now, put beads, nanobeads into the channels, and look at the deformation. So you have like actin stained cells here that move through the channel, and then we can quantify here the movement of the beads, and so you can see one slipping through here, and the other one here. So you can really see without seeing the cell. You can see the movement here. Yeah.
Is this is this buckling? Is this crisis because the gels keep swelling once it's casted? Or?
Yeah. So that's another big problem. I think that's another half year because when you do polyacrylamide gels and keep them, they're swelling. So whenever you define the size of the spacers, you have to calculate in the few percent swelling of the whole gel. It's very annoying, but
Jesus, yeah.
Yeah.
To the force.
Yes, but at least now we can look at cells that are moving here like through, and so you can see the beads moving apart, and we can cross-correlate what is happening to the actin and the beads. And actually here you can see an example where the cell moves through the constriction here, and you can see the actin burst here while the beads are moved apart. But yeah, we have to do way more quantifications on that. And these are our first movies now where we can see actually microtubules and actin. And I think this is quite satisfying now to see that it's not properly quantified yet, but so whenever the mTOC moves through the constriction here, you see the beads moving apart. And here's another one where you can see a cell that actually moves. This one moves nucleus first, but you can see, here the mTOC, and we can now measure when are the beads moving outward while the cell is moving through. But yeah, it's early days, but at least now we can do it. And
Non-adhesive or adhesive?
polyacrylamide is so we can make it. This is non-adhesive. But the problem is, at the moment, we still have it on slides that are adhesive. So we can't put it on inert slides so that we can get it completely non-adhesive. The problem is, if you have it completely non-adhesive, the cells don't even go in. They have a problem invading. So the first step of going into the channel is highly adhesion dependent because it's very asymmetric. Once they're in, they can move without adhesions. So now we do channels, basically we put them on substrates where the entrance is actually adhesive at the bottom, and then it gets non-adhesive. But this is all doable because we just take the gel and put it onto a pattern surface. Yeah?
Remain within the
this year.
Is that like just a side effect, or there's a reason for it?
This happens way more when you go to soft substrates. So in a stiff channel, this usually doesn't happen. So they leave a lot behind, yes. But that's all I can say, that it happens. Why and when and so on, I can't tell you. Yeah. Uh-huh.
Since it doesn't happen on the stiff channel, so it's just a side effect of the material of the gel, or?
It doesn't happen if you have an open channel, so you really need a constriction at the same time. if it's now due to the material, like it's pretty inert for the cells. If you put a cell on polyacrylamide, it's absolutely non-adhesive. So I don't know why they do it. It's good question. Yeah. So basically, that's it. What I wanted to show you. And all very preliminary. So this is the lab here. They are not always as happy as they look here. So especially this one, he did the the soft channels. That's Mario. And the first part with the Doc8 was mainly done by Patricia, but also with a lot of support by others in the lab. So the polarity, mesenchymal versus amoeboid polarity, was done by Nika, other PhD student in the lab. And yeah, we always have a lot of support by our facilities here at the Institute in Klosterneuburg. And yeah, a lot of collaborators and of course our funders. So thank you very much.
Very nice. Thank you very much. Questions? Think about it, because so why does a GEF locate to the constriction? How does a GEF know where the constriction is? What's the signal?
Yeah, what's upstream actually? So we know one thing that is upstream. It's hippo signaling actually. So if you knock out MST1 and 2, get the same. You get a phenocopy of the Doc8 knockout. This has also been mainly shown in T cells. So there's a parallel development by Judith Mandl at McGill, looked at the Doc8 knockout T cells. And there, if you knock out MST1, you get a phenocopy of the Doc8. We in dendritic cells had to knock out MST1 and MST2, and then the cells have a few more problems. But still, we lose this central actin structure. So hippo signaling is upstream. Then the question is, what is upstream of hippo signaling? What's the force sensor? And I doubt that we will ever find one. Pretty sure it's not piezo. So I guess it's actin itself. That is always my guess, that it's kind of a feedback. Yeah.
Okay. Cool.
So at the beginning you had this forest of pillars, and you showed the nucleus is the one that leads the whole thing and that probes the space. And then later you had the confinement assay where the nucleus lags behind, and the mTOR is the decisive pass, or I don't know, not pass finding in the same sense. How can you bring those two observations together?
So basically, when also in the previous work, when we showed that the nucleus is decisive, Jörg only found that the nucleus is decisive if the nucleus is at the front. In the cells that moved the other way around with the mTOR at the front, then it was not the mTOR was not so they were not pore selective anymore. So I think it really would make sense that they, when it's difficult for them and it's getting narrow, they move with the mTOR at the front, and then it's primarily about dilating something that already exists. How they choose then what to dilate, I don't know. I would be my guess would be that they're very much sending the rigidity again, that they push into it and that they go for the soft path. But in general, so this nucleus first configuration is when things are really going easy for the cell. So when there's not enough counterforce for the nucleus to sweep it to the back, then it lands at the front and then it just slips through the gaps. That's at least the idea. It's very, very passive model. Yeah.
So we've seen that when a cell enters these stiff channels, that we see this actin structure appearing there.
Okay.
Have you ever checked? No, I mean in the videos in your talk. And when you look at cases where the cell retracts again and doesn't go through the channel, how far does this actin structure get? Is there a specific point where the cell says, okay, now I'm going through?
A checkpoint when it decides to pass. Yeah, that's a good question.
Or do they always go through in your cases?
So as far as I remember, when the mTOR is through, that's basically it. So they don't turn around anymore. If there's a checkpoint that is before that, I don't, the nucleus is not the checkpoint. So if the nucleus is through and the mTOR is still behind it, they often turn around. So
But the mTOR is more decisive.
We also looked a bit at neutrophils, which are interesting because they have this multilobular nucleus, and then they often, if you put them through a wide channel, then they put part of their nucleus into one and part of the nucleus into the other channel, and then the side with the mTOR always wins, and then they retract the other part of the nucleus.
Okay, that's interesting.
So I think I would say microtubules are really important here.
Okay, thanks.
Hello.
Yes.
Sorry, I might have missed it, but when you talked about the microtubules pushing in the central region, is it a active push via polymerization, or is it a passive push because the stiff microtubules have to go through the channel?
My guess would be rather the second, so that they're kind of a counter force, a compression insensitive element, because they have such a long persistence length, though, that it's like a ratchet. The actin is kind of pushing on the microtubules. A bit like neurons are doing that, right? So if you get rid of actin, they have actually the axons are even growing better, and that's just driven forward by microtubules. So in this sense, we are just saying, yeah, every cell is actually partially using this typical neuron or glia. Glia cells also have it. It's actually all the cells in the brain, which is interesting because, like in the brain, it doesn't make a lot of sense to pull on things because it's so soft. So maybe everything is based on pushing. And yeah, you even showed that neurons go through softer, like they even actively move through to softer areas, right? So yeah.
Just move our way back.
Okay, the osactin structure, how general is it? You find that in any cell types?
Immune cells, yes. Not in neutrophils. Actually, neutrophils don't even have DOCK8. And we also couldn't find, and Judith Mandl, who also looked at it, couldn't find the structure. Maybe because they don't even have a significant nucleus that they have to kind of protect. In non-hematopoietic cells, so DOCK8 is in a few brain cells, but it's primarily a hematopoietic protein. It's actually a well-known protein. There's a congenital immunodeficiency. Kids that lack DOCK8, they have lots of problems. So yes, that's immune cell typical. If this structure has some analogous structure in other cells, in mesenchymal cells, I don't know.
This was actually something I was going to ask anyway. So if this is specific to phagocytotic cells, because when I had to work with AFM with phagocytotic cells long time ago, whenever you touch them with the cantilevers, they start pushing. But only in phagocytotic cells, not in fibroblasts, not in neurons. So that's why I was wondering if
T cells also have it, and they are not phagocytic.
Okay.
So it seems to be like, I would say, amoeboid cells. This thing that if you poke a cell, that it pushes back, that is something I think is probably universal in immune cells. Also fibroblasts, it has actually shown by people, if they put with a cantilever into a fibroblast, there's WASP that is triggered, and then it pushes outward. Yes. Yeah, it's much slower. Yeah, these cells are in general much faster. Yeah.
Okay, in the beginning of your talk, probably in the first slide, you have showed probably the cells like to avoid the dense network. But if you keep these cells in front of a narrow channel where the width will be probably 4 micron, and if you take the same cells in front of another narrow channel where the width will be probably 10 micron, then which the narrow channel cells will like?
They rather, if they move with this nucleus first, they rather move through the bigger one.
Not the narrow channel.
Sorry?
So they don't like constriction so much.
Then if there's enough, if you give them a choice, we made assays where you have one channel and then you have 4 different pores to choose, and they always choose the biggest one if they are with the nucleus first. If they're in the other configuration, they don't care about the size. Then I would say they dilate it.
Thank you.
So you said that in the ACNA knockouts, the actin burst is not there. So I guess you also checked whether the DOC8 is not localizing.
Yeah, we have to look at this. No, we didn't do that. That's pretty preliminary here.
You would expect it. So
yes.
then in evaluation question, I don't know, did you activate the channels so to see whether any integrins are involved there, or look at GFP, oxalin, or something like that?
We cannot see adhesions in these channels. Basically, there's not enough of a challenge. We get adhesions when we let them move through a constriction and they get stuck at the back. Then they have to pull at the front. And then we see some adhesions, like you do, right? This is a situation where maybe there's so much, they have to fight so much to move the leading edge forward and against the backward pulling of the nucleus that they then start to anchor. We also did some assays where we pulled on the cells with magnetic tweezers.
Yeah.
So we let them swallow beads, and then we pulled on the back and then see if they make more adhesions. So we see that they become more dependent. We saw that they become more dependent on adhesions, but we were never able to properly visualize the adhesions. Yeah.
But I mean, you're trying to visualize any enrichment of any adhesion proteins in this actin burst zone, so not in the front. So not visualizing proper focal adhesions or anything like that, but some kind of an enrichment. You could still visualize it, I guess. No?
Yeah, we looked a bit at VWASP.
Yeah.
So, but in these channels, it's difficult to quantify. Yeah. We never managed to do that. But my guess would be if they get stuck at the back, they really make more adhesions. Because otherwise they couldn't get so long. Yeah.
Thank you.
Yeah, I was just thinking, could you even speculate why would nature do that? Why would you have two, so to say, populations, one with the nucleus in front and one in the back? What would be a benefit in tissues? What is a benefit for the immune cells to basically divide in these two populations?
To have this, to push outwards and to push forward at the same time, or to have these two force components.
No, I mean, you said to yourself, right, some cells have nucleus in front and some cells have it at the back, right?
Okay.
And that is going to define how they behave, can they squeeze in, how fast they are,
yes.
Right? So why would, and that seems kind of random, right? Some cells.
No, it's really dependent on the environment. If the environment is permissive enough, they just, the myosin at the back kind of wins in pushing forward the nucleus. And then you end up with this nucleus first configuration, and then they kind of slip through the biggest hole that is there. If the environment becomes more restraining, so if the cells have to push their nucleus through the pores, then they just inherently end up in this, having the microtubule organizing center more at the front, and then pushing outwards. So I don't think there's much of a that's just a convenient way how the machine works. I would say I wouldn't interpret too much strategy into it. It's kind of to me these cells are just off-road vehicles that can solve any type of problem in a very plastic and adaptive way, and that's just the convenient product of the nucleus being flushed to the back that they then are able to push outwards. Not sure if I answered your question, but
yeah,
maybe.
True. I mean, I just understood it because you said, right, once they come to at the very beginning, right, when they were coming to the small
constriction.
Constrictions, they decided if they go through or not, depending if they had nucleus in front or not.
Yeah.
So they were kind of.
I think it's a mistake that I said, my mistake that I said decide, because I don't decide. It just happens that the nucleus gets flushed to the back, or that they are in a configuration where they maybe are a bit slower and they end up with having the mTOC at the front and the nucleus on the back. It's just it's an enormous variety that you have in cells. I also wouldn't. to me, if we have a population of cells, every cell to a certain degree, it's an individual. First, because of its history, that maybe it ends up in a bit different configuration, but also because of its molecular composition. So it just has a normal distribution of every protein. So every cell is a bit different. And I personally think that also every cell has probably a bit different talent to solve a problem. So we're trying to put these cells actually now in heterogeneous environments, and then see if there are some cells that do better in one versus the other environment. And I'm pretty sure there are like more multitaskers or more single strategy cells, because they are just inherently noisy. It's not exactly one cell type. Even if you have immortalized cells, they are not all the same if you measure their protein distribution.
I would be curious in asking a question whether there is a cost of putting the centrosome before the nucleus, like in terms of speed or like length they can travel. After all, they have to go to a certain focus and then act there. So like depending on the mode of the migration, whether nucleus is first or centrosome first, could you comment on that?
I ask really many times people in the lab to measure how is this configuration mTOC first versus nuclear first, that we cross-correlate with speed of the single cell in the same environment. And you just have to, we didn't get enough data to have really a significant difference. But my guess would be, whenever you have a cell that is slower, then it has a higher likelihood that the mTOC is at the front. Maybe these are also the cells that have a bit of bigger nucleus, so that it gets more hold back, right?
Because in the video you showed in the beginning, the cells that faced the constriction early on, they would not make it as fast, right? So I mean, they would also stop at some point. I mean, that's maybe
they're definitely, once they're in this situation that they're more constrained, then they are slower. Yeah. And they move more with the mTOC. But if you have same conditions and look at cells, some cells might be with the mTOC first, some with the nucleus first. I would guess that it's the slower ones that have the mTOC at the front.
Thanks.
Have you tried a sequence of constrictions to see if the configuration is maintained over the...
Yeah. Yes, we tried multiple constriction also to see if the cells can learn and things like that. We now also don't have really good quantitative data on that, but it is pretty clear that if you have one constriction, they have a tendency that they move more mTOC first, and then they move through the second constriction with the mTOC first. So it's kind of a selector for mTOC first. So that is a truth. But yeah, we also wanted to see if they get better and better with it, but that we didn't achieve yet. Yeah.
Okay, thank you. So because the seminar room is not large enough for you, we also have an online audience. There's an online question. Do we want to have this first? Should I read it, or can the person speak? I can read the question. Did they write it? Okay, so we give the person a minute to write down the question, and I think Jona raised his hand in the meantime.
So thanks a lot. And first of all, I think you can tell Mario that was like worth all the effort, because that's exactly what I was thinking about.
Mm-hmm.
And
I will tell him.
I guess this is already implicit, right? That what you're probing with this is the invasive potential for these immune cells, right?
Yeah.
Now, could you ask, like, one of the, or potentially even answer one of the most fundamental questions that I'm asking myself right now is, if we activate T cells, what does that do to the invasive potential? Because that's what we're trying in CAR T cells, et cetera, like just make them super, super aggressive active.
Yeah.
Does that also make them infiltrate better, or maybe even not? And that would be the perfect assay to do that with.
Yeah, we didn't do too much with T cell invasion, because like we very much like cells that we can somehow align. With T cells, that's a bit tricky because they are not. They are more random movers. But my guess would be that the T cell blast is better than a naive T cell. But if you have them, like, if they are more activated, like by a stiffer substrate or so, if they are then able to move into stiffer regions, that would be quite interesting, I think.
I was thinking via the T cell receptor, right? So we have this image, right? T cell arrives at the tumor site and gets activated because it recognizes the antigen, and then starts killing. And that's great. But if that inhibits the migratory or the invasive potential of the T cell, it's just going to kill on the outside. And maybe could be one of the factors why T cell therapy does not work so well in solid tumors. They just can't get in.
T cells, whenever they get a T cell receptor signal, they stop moving. So that is shown. So basically, they have to stop to kill also. If they then killed and go to the next one, how that changes, no idea.
Exactly
it would be interesting to know. Yeah. I think Philippe Busson did a few essays like this. So he's at Pasteur. He had a few relation between the T cell receptor stop signal and then the migratory capability. I can't exactly remember.
But with constraints, like with these mechanical constraints, but free, I can totally see
he did
how
free
relevant
migration.
that is then for dense tumor environment.
Yeah, it would be really also interesting to get dendritic cells from a stiff tumor, prime T cells, and then see if the T cells are better in moving into something stiff. If there's a mechanical memory over one signal cascade.
So TIL therapy with this would
would be interesting to measure it. All
right, so there are a couple of questions. First question by Wolfgang Goldman here. He asked if it's only actin around the nucleus that is responsible for squeezing the nucleus when passing like gaps, or if microtubules also matter for
the actin is not directly around the nucleus, it's more in front of the nucleus with the mTOC. And yes, we think microtubules are also important, but I cannot tell exactly how important, so what they are precisely doing. My favorite idea is that it's a mechanical function.
There's two questions regarding to cancer. So is this relevant? How relevant is this to cancer research? Or the other question is, if I understand it correctly, does the stiffness of the collagen matter? By tweaking collagen backbone, can you change metastasis potential?
Yes. So in general, on cancer, so my justification for this type of research is always like, if we find out how a cancer cell moves is pretty difficult because they are inherently an accident cancer cells. So they are very inefficient in what they're doing. So it's difficult enough to find out how a professional cell moves. But once we know that, we can infer on what the cancer cells are doing. So that's a very basic science argument. And there are many cancer cells that have amoeboid configuration. And so, But I find it a bit frustrating to work on cancer cells because they do what they're doing so bad. And then the second thing, of course, for T-cell therapy, as you just said, CAR T-cells, I get a lot of questions from the clinicians that say, how can we make the CAR T-cells invade better? So I don't have an answer, but I can always refer to what we find out, and maybe one can translate it to this T-cells.
Yes. The last online question is basically, I think, if you have worked on like parasites already. If you have tried this with parasites and if they show similar behaviors as eukaryotic cells.
Yes, we work a bit, like we have dictyostelium in the lab. So cool. I did I like to watch amoeba under the microscope, which are very similar to leukocytes. But we not doing that professionally. There are a few labs that are now really good in basically asking phylogenetic questions about movement. And so Lillian Fritz Leilin is probably the most prominent lab working on that. It's really, really great work. And actually you'll find a lot conserved mechanisms, but you find every variant of it in protozoa do crazy things. So some of them, actually, they swim with a flagella, and then you squish them a bit, you confine them, and then they start to crawl. And they do that, some of them do it with actin, some do it actually by microtubules. So, but this transition became very popular in the protozoa world, actually, this, like, upon confinement, that the cells can basically start crawling.